All right, good morning, everybody. I apologize, my voice is a little rough today, so bear with me as I record this first part about enzymes. So first of all, there are updates to the schedule, right, that has been posted on Moodle, and you can see the reflected updates. Most notably, I have added an extra study session for this unit content, simply because we only have 4 lecture periods before the next exam, and I thought we needed more time to review if you wanted it. All right, so the exam results, I will pass these back on Thursday. The class average was about an 83%, so that was awesome. That's 2% higher than it was last year, so great job there. I would say if you got a 35 or higher, great. That's awesome. Don't worry about your score. If you got below that, particularly if you didn't do so well or not as well as you wanted to, that's okay. This is the first exam. There are still plenty points available left to obtain this semester. Think about how you studied and how you might want to study a little bit better for next time. And if you want, you can go ahead and meet with me. I'm happy to meet and talk about what went well and what we can do better. There is an opportunity for points back. I will change the due date on this slide. But essentially, you're going to get an opportunity to, if you have gotten any one of these wrong, you can correct it for either full points back or half points back. I will talk more about this on Thursday when I actually pass back your exams. So let's dive into the lecture. All right, so before the exam, we did a review of energy. So I put in this slide of key terms that we went over. I'm not going to go over all these terms again. If you need a review, go ahead and watch that video from before the exam. But we left off on this question here, right? Now I'm going to leave you in suspense in this question, and I'm going to flip ahead to the next slide because we will review these questions on Thursday. All right, so let's talk about enzymes. All right, so we talked about reactions and how they can be spontaneous, so they don't need an energy input. But just because they're spontaneous does not mean it's necessarily a fast reaction. All right, so there are ways that we can speed up a reaction, right? We would say a catalyst is any agent that speeds up the rate of a chemical reaction without being consumed within the reaction. All right, so it's something that speeds up the reaction, but we're not actually consuming it. It's not being used up entirely by the reaction. And enzymes, are protein catalysts in our living cells. So enzymes are a type of catalyst, right? Enzymes are proteins, and enzymes increase the rate of a chemical reaction. All right, enzymes themselves, they do not get used up during this process. They can be used over and over and over again. All right, so because an enzyme is a protein, right, and we know for proteins, structure is everything. Let's look at the structure of an enzyme. All right, if we're looking at an enzyme, one of the most important parts that you'll have to know is that an enzyme has an active site. Right? This is a site within the enzyme where the reaction takes place. It's typically a groove, or it's typically drawn as a groove. We can see in this image down here, we have this active site, right? It looks like this opening, this groove within our enzyme. Our substrate will then go into the active site. And we can see in the next picture here, our substrate is within the active site. It's bound in place. And we would call this the enzyme substrate complex, right? When we have both the enzyme and the protein, and the molecule, the substrate, bound together, right? When they're together in this area, then the reaction can take place. All right, so the substrate is any reactant that will bind to the active site. All right, any type of molecule that's going to go through this process and use an enzyme to help it get to its products. Now, it's important to remember enzymes, they are highly specific to their substrate, right? Notice this active site. This active site is specifically for one particular type of substrate, all right? So that means if a different type of substrate tried to bind with this active site, it would not work. So it has this high specificity, right? And again, we know that enzymes are a protein, right? And structure determines function. So the structure of this active site will determine which substrate can actually bind, right? And which function will take place. All right. I want you now to pause this video and watch this TEDx talk about enzymes and how enzymes can help reactions take place. I'm not going to record it here because if I do and upload it to YouTube, I will get flagged for copyright infringement. But please watch this video now. All right, so hopefully you watch that video. Let's talk about a few other terms about enzymes. All right, so how do enzymes work? Well, enzymes allow molecules to get close enough that they can actually cause some sort of a bond rearrangement, right? Now, in order for a reaction to occur, to start a reaction, we need an input of a little bit of energy, right? And this is called activation energy, the initial input of energy to start a reaction. All right, if we look at this graph here, we have the progress of the reaction along the x-axis here and the free energy along the y-axis. Our reactants here are going to turn into our products, right? And notice that we have a delta G, right, or Gibbs free energy less than 0, so this should happen spontaneously. We don't need a huge input of energy from, say, ATP to make this happen. Right, but we do need a little bit of activation energy, right? So that's different than energy needed for a non-spontaneous reaction. This is just the activation energy where these molecules can reach this what's called a transition state, right? So we have to get these molecules close enough so the bonds can be rearranged. Now remember, all these molecules, they have electrons encircling all of the atoms within the molecules. Those electrons are negatively charged, right? And like charges repel each other, right? So we have to get these molecules close enough that bonds can rearrange, right? And that's not easy to do because those electrons repel each other, which is why we need this initial input of energy, this activation energy, to get it into this transition state. Once it's in this transition state, right, the bonds can be stretched, they can be broken, they can be rearranged. All right, there are some common ways to achieve this activation energy, to overcome this activation energy. One of which is large amounts of heat, right? So an input of heat can give us this activation energy that we need. But another one is using enzymes. All right, enzymes are able to lower the activation energy needed for a reaction to take place. So by using an enzyme, this reaction can take place a lot more quickly and a lot more efficiently. All right, so how do enzymes lower activation energy? Right, so that video mentioned a few ways and gave us some helpful analogies and demonstrations. But let's talk about a few. All right, so one way that enzymes can lower activation energy is simply by straining the bonds in a reactant to make it easier to achieve a transition state. All right, so if we have our substrate going into our active site here, this active site, once the substrate is bound, can strain the bonds within that molecule, right? And by straining those bonds, right, it makes it easier for those molecules to overcome this transition state that it needs to get to, right? Another way is just simply orientation, right? Let's position these reactants close enough together that bonding can actually happen, right? So here we have an enzyme, and we see two different substrates are going to bind to the active site, right? Once they're bound to the active site, right, they're close enough together that bond rearrangement can occur, right? And that's good. We want that bond rearrangement, right? And we want these molecules to be close enough because, again, those electrons are going to try to repel each other. So we can position them nice and close together by using an enzyme. Now, enzymes can also change the local environment of a reaction, right? So maybe there is a changing of an environment by temporarily bonding the enzyme to the reactants, right? Something is changing about this local environment to make this process, to make this chemical reaction happen more efficiently. All right, let's talk about how enzymes themselves are affected by the environment. All right, so enzymes can affect the local environment of a substrate, but the environment that the enzymes are in can also be affected. All right, so most enzymes are effective within a very narrow range of temperature and pH, right? And this makes sense, right? Because structure determines function. And if the enzymes are very specific to the function that they do, right? They only perform one function and they can perform that function over and over and over. It makes sense that they are only able to perform these functions in a very narrow range of environment. All right, so if we look at these graphs here, right, we have temperature along the x-axis in the first graph, and the relative chitinase activity. All right, so chitinase being the enzyme here. Now, if we took that enzyme that lived in a bacteria that lived in a cool and neutral environment, right, so this blue line here, we would see that peak enzyme activity actually occurs around this 45 degrees Celsius range here. Right, but if that came from a bacteria that lived in a hot and acidic environment here, we see that its peak enzyme activity is actually around 60 degrees. Right? So depending on the organism and the environment that the organism is adapted to, right, can affect how an enzyme functions. Same with pH. All right, so if we have enzymes in different pH, again, if we have a bacteria that lives in a hot and acidic environment, right, this enzyme is going to be more effective at more acidic pHs, right? So like a pH of like 2.5 or 3. Now, if we have this enzyme coming from a very neutral environment, right, we can see that this enzyme is most effective around that neutral number of around 7 there. All right, so we'll go over this group question in class here. But let's talk about the last couple of slides that I want you to be aware of for enzymes. All right, so enzymes have that active site that is very specific to their substrate. But that does not mean other molecules cannot bind. Right? In fact, we can have things called inhibitors. A couple of types of reversible inhibitors here are a competitive inhibitor. So those are molecules that bind to the active site themselves. So it's a molecule other than the substrate. So we have our substrate here, and we have our competitive inhibitor. If this inhibitor binds to the active site instead of the substrate can no longer bind. All right, so that means that this enzyme is no longer functional for the reaction that it needs to perform, right? Because the substrate can no longer. A competitive inhibitor is in the area where this enzyme is. We need more substrate to try to combat that competitive inhibition. There's another type of a reversible inhibitor called a non-competitive inhibitor. And this is where the inhibiting molecule does not bind to the active site, but rather binds to this allosteric site. Right? When it binds to this allosteric site, it changes the shape of the active site so that the substrate can no longer bind, right? Because shape is function. And if this non-competitive inhibitor, right, changes the shape of the active site, that active site is no longer suited for the substrate to come in and bind. And therefore, their reaction cannot take place. Now, these types of inhibition are reversible, right? Meaning that they are not permanent. Once the inhibitor leaves the enzyme, right, the enzyme is then free to go about its normal function, right? Then it can function normally, it can bind to the substrate once again. There are irreversible inhibitors, right? And so these are inhibitors that will bind covalently to an enzyme to inhibit function. And once it binds to that enzyme, it permanently blocks the active site, right? That enzyme can no longer be functional, right? This is not a reversible reaction. So, a good example of an irreversible inhibitor is actually nerve gas. Nerve gas binds covalently to its enzyme, and once this occurs, right, it's not reversible. So, neurons and muscles no longer can communicate, right? And this is eventually going to cause death. So, cells don't typically use irreversible inhibitors, right? In fact, nerve gas is one of those types of biological warfare that's prohibited, right? So, cells will normally use a reversible inhibitor instead, right? So, if they need to inhibit the function of an enzyme, right, maybe they need to stop a reaction from occurring, then cells will use a reversible inhibitor until it once again needs to make that enzyme available and run the reaction. All right, I am now going to switch over to my lecture from last year to talk about this overview of cellular respiration.
We are starting content for Unit 2. Unit 2 is focused on 3 topics: cellular respiration, fermentation, and photosynthesis. Right? It's only 3 topics, but there's a lot of steps in each of these processes. All right? So it might be beneficial for you, it might not be beneficial for you. I like this unit because I can study better when I think in terms of processes rather than isolated biological terms. We're going to start with cellular respiration. Right? So today we're going to go over some concepts we need to understand before we dive into cellular respiration, as well as just the broad overview of cellular respiration. And we're going to start on the first stage of cellular respiration. So if we look at big overview here, we're talking about metabolism, right? So metabolism is all of those life-sustaining chemical reactions in a cell, right? A whole host of reactions, a lot of reactions. We look at this diagram. I like this diagram because it shows you just how complex it can become, right? Big picture here is that we are converting food energy, so what we're ingesting, into cellular energy, right? Energy like ATP. And it's a multi-step process. It's not straightforward. It's not you eat food, you get energy. There's a lot of steps in between. And again, I like this image because they even color coordinated it to what types of molecules you might be ingesting. Like carbohydrate metabolism is different than lipid metabolism, is different from amino acid metabolism, right? So, whole host of chemical pathways. We're just going to brush the surface. We're just going to focus on glucose, which is part of the carbohydrate metabolism here. All right, so if we look at metabolism, like I said, there are so many reactions in metabolism, but we can categorize reactions into 2 different pathways. Either they're going to be catabolic or they're going to be anabolic. Right? These are very similar to exergonic and endergonic reactions. Right? So catabolic reactions, they are exergonic, which means they release energy. They happen spontaneously. But we're breaking down cellular components. We are taking a larger molecule, breaking it down into its component building blocks, releasing energy in the meantime. Right, so if we look at our image up here, we have our one large molecule. We're breaking it down into its smaller units. We're releasing energy. Right? And again, this is exergonic. It happens spontaneously. Right, so that delta G is negative. Thinking back to our Unit 1 content. versus an anabolic pathway, right? Completely opposite. Instead of breaking down a large molecule, now we are building up a larger molecule. So in animalism, we have smaller units, right? Maybe these are all monomers that we're going to combine together to create a polymer or a larger molecule. This is not exergonic, it's endergonic, which means we need an energy source. So typically, we are going to couple energy with this reaction. So we need energy in order for this to occur. So this is important when we think about synthesizing different components for our bodies. Maybe we're building up a carbohydrate molecule. Right? Maybe we're building up glycogen to store energy. Maybe we're making a protein, so we have to combine a bunch of amino acids together. Now it's important to note, for all of these pathways, there is going to be an enzyme coordinating each step. All right? Even if that pathway is catabolic, even if it's exergonic, so it happens spontaneously, there's going to be an enzyme every step of the way to make this go faster. Right? So if you look at metabolism, you could have hundreds to thousands of different enzymes involved here. And we'll see a few here and there as we talk about cellular respiration. All right, so I want to just kind of elaborate on these two topics just a little bit more. I really reinforce catabolic versus anabolic. So if we look at anabolic reactions, right, we are making a larger molecule. So we might not get the molecule that we need from food, right? But we can take the components that we've taken in from our food to build up the larger molecule that we do need. Right? So for example, if we look at a protein, right, the smaller units are our monomers, right, are our amino acids. Those amino acids will come together, right, to form our polymer of our protein. Because we're building it up, this reaction doesn't just happen spontaneously. We need an energy input, so we need to use energy in this process. Now, the energy that we typically use is ATP, which we've talked a little bit about, but there's another molecule that we will talk about today called NADH, which is also an energy carrier molecule. All right, so anabolic reactions, big picture. Needs energy, we are building larger molecules. All right, so the opposite of that would be our catabolic reactions, right? So in this case, we're breaking down molecules. Maybe we ate a potato or something, right? Something really starchy. We've ingested some starches. We need to break that starch down into its component parts. Right? And the component part of a starch is sugar molecules. Once we've broken it down into its smaller units, those smaller units can then be used elsewhere. I like this image because it kind of shows you different pathways that this can occur. So we might intake our food, let's say that's a complex carbohydrate here, right? So like a starch. That starch can be broken down into glucose. Right? All that breaking down is our catabolic reactions. So all the arrows here that are like this green, this teal green in color, those are our catabolic reactions. Now once we have those smaller units, right, so once we have all of those glucose molecules, they can be used in anabolic reactions to build up molecules that our body needs. Right, so we have our building blocks. We're now going to use our building blocks to produce the molecules that our bodies specifically need. Now, in catabolic reactions, we don't need energy, right? But we produce energy. Right? And so our energy produced, again, is going to be in the form of ATP or NADH. A lot of different pathways, again, depending on the type of food we are ingesting. Now I have one more image on the slide related to, or the next slide, related to catabolic and anabolic reactions that really simplifies things. So we're taking in food, we're breaking it down, we're using those components that we have broken down to build up other molecules. The slide is just really simple, shows you a broad overview here, not too many complexities. See, when we break them down, we're releasing energy. When we're building up larger molecules, we're using that energy. Right? So they all kind of interplay with one another. All right. Let's do a group question. I want you to tell me of these two reactions that you see, which one is anabolic and which one is catabolic. I know we've talked about ATP, but because cellular respiration is all about producing ATP, it's worth coming back to and talking about it in a little bit more detail. I noticed this mistake in my last class. I grabbed this picture from the internet thinking, oh, I really like the simplicity of it. This picture has a mistake on it. Does anybody see the mistake? I do know. If you look at the molecule here, it says that this sugar is a ribosome. It's not a ribosome, right? So if we look at ATP, it's really similar to an RNA nucleotide in that it has a ribulose sugar, not a ribosome, a ribulose sugar connected to a base. And that base is going to be adenosine, connected to 3 phosphate groups instead of only 1 that we see in RNA. All right. So this image I did not look closely enough at. Ribosome is wrong. We know that's an organelle. It should be ribulose, or ribulose sugar. All right. So it's really similar to an RNA nucleotide in its structure. The big thing here is that it has 3 phosphate groups, and that's key. So these phosphate groups, they have a lot of negative charges. Right? And we're trying to force 3 of these together. And in fact, we have forced 3 of these together. They are bonded together in this triphosphate part of our ATP. Okay? Now we know like charges repel each other, and we have now forced these like charges to bond really close to each other. What that means is that those bonds have a lot of potential energy. Those phosphate groups naturally want to repel each other. Right? So we have a lot of potential energy in those bonds. When those bonds are broken, we release that energy. And that is the energy that we actually use to power a lot of our cellular functions. Right? So energy is stored in those phosphate group bonds. Right? Now when we take away a phosphate group, right, via hydrolysis, right, we release energy, right, we release a phosphate group, and what is left is our ADP molecule, adenosine diphosphate. Di meaning 2, because now we only have 2 phosphate molecules. Okay? This one does not carry as much energy. Right? In order for ADP to be utilized, we have to add another phosphate group, which involves energy. So we have to add an energy, add in a phosphate group to bring it back to ATP. Okay? But the big picture is that ATP is not really used up. It's just part of this cycle. ATP, to ADP, to ATP, to ADP. Right? It just cycles. Those 3 phosphate groups are really key. All right, so we have ATP, but we also have NADH. NADH is also a molecule produced in cellular respiration that carries energy, right? But it does this in a different way. So in ATP, that energy is held in those bonds between the phosphate groups. In NADH, this molecule carries energy. It carries energy in the form of electrons and protons. Right? So it's not bound in a bond, but rather it's energy in the form of it being able to carry electrons and protons. Right? So we would call it an energy intermediate, in that it can accept electrons and protons removed from other molecules. So NADH is our one on our right-hand side here. Right? If we look at it carrying these electrons, carrying these protons, we see that show up on the top of our molecule. We see this one has 2 H's. It has a ring of carbon with only 2 double bonds, and it has a nitrogen with no charge. Right? So this one is currently carrying energy. When NADH releases that energy, releases the protons, releases the electrons, it turns into NAD+. Right? And again, the change here occurs where we only now have 1 hydrogen. Instead of 2 double bonds, we have 3 double bonds in this ring of carbon. And our nitrogen has a positive charge. Right? But the molecular structure, that is what is physically changing between NAD+ and NADH. Now, this molecule is useful in that it carries the energy in the form of electrons and protons, and then can donate that energy in the form of electrons and protons. Now, NADH is one of those that I feel like you probably haven't heard about as much. A lot of the focus is typically on ATP. So I just want to have it reiterated to you. Right. So the next slide here is just a review. I want you to review how do each of these molecules provide energy, right? And then think back to our last unit. What organelle is going to play an important role in cellular respiration, which is the topic we're heading into? All right, one concept we have to go over before we really dive into cellular respiration is redox reactions. Right? Again, this is not a chemistry class, so we're going to stick to surface level knowledge here. Right? And honestly, redox reactions are not my favorite thing to teach because I think it feels clunky to me. But nonetheless, we are going to learn redox at a very surface level here. So these are reactions involving electrons being transferred, right? So NADH, we know it carries electrons and can transfer electrons. This is involved in a redox reaction. When a molecule is oxidized or goes through reduction, right, we're removing electrons. Did I say that wrong? I think I might have. Oh. So, oxidation. Removal of electrons, right? Reduction, on the other hand, right, is the addition of electrons, which that definition is a little counterintuitive, right? If I'm reducing something, it doesn't seem like I should be adding something, but what I'm reducing is the charge, right? I am adding electrons, which have a negative charge, so I'm reducing the overall charge of that molecule, of that atom. Right? So if we look at our example here, I have molecule A and B. Molecule A has an extra electron. It's going to transfer it to B. So you would say A is oxidized because it's losing its electron, and B is reduced because it's gaining an electron. And the mnemonic that I always have to repeat in my head to remember this is oil rig, right? Oxidation is loss. Reduction is gain. And we're talking about electrons. All right, so we're going to go from talking about this simple redox reaction. We're going to scale it up quite quickly. So my next slide here, we're going to talk about the overall equation of cellular respiration. All right, so in cellular respiration, we're taking a glucose molecule and an oxygen molecule, and we're turning it into CO2, water, and ATP. Right. Now, if we're just focusing on redox reactions, right? Often hydrogen ions are going to accompany those electrons. Okay? So if we look at glucose, glucose is losing hydrogen to become carbon dioxide. So we can say glucose is being oxidized. Versus oxygen, on the other hand, is going to gain some of those hydrogen ions to become water. So this would be reduced. Now we could look at this in a different way to see specifically how the electrons change, which is what the second image is all about. Same reaction, just a different way of looking at it. And here what this image is emphasizing is that not only are we losing and gaining electrons, right, but that can change the nature of a molecule, right? So if we look at glucose, we have the CH bond. This is a non-polar covalent bond. Electrons are shared equally. But when we lose these electrons and transfer the hydrogens, right, when it becomes CO2, those oxygens are now held more closely to oxygen, and it's part of a polar covalent bond. So these electronegativities can change as well, right? But I want you to really focus on big picture here, right? I want you to look at the removal and the acceptance of electrons to know which is being reduced, which is being oxidized. And I think the best way to do this is practice. Right? I thought I had my practice slide next. I don't. I also put in the slide of NADH. Not that I would expect you to know this specifically. Right? What I want you to know about NADH is it's a carrier electron. But if you're curious to see specifically how it carries it, we look at this nicotinamide, I always have trouble saying this word, molecule, part of the molecule up here, right? And it can be reduced and oxidized back and forth, and it's all about those two electrons, those two hydrogens. All right, now let's practice. So I want you to look at this equation and try to figure out which of the reactants, right? So first part of the equation is being reduced and which are being oxidized. Give it some thoughts, and then we will go over this together. I'm going to make that part available afterwards, so you don't have to write down everything. So let's go on to metabolism again. So we have all of these pathways. Right? A lot of different redox reactions, a lot of anabolic, a lot of catabolic reactions. The cell has ways to regulate which of these pathways actually happen and which don't. Right? One way is by simply turning genes on or off. Right? So this would be taking DNA, transcribing it, translating it to a protein. Right? And the cell can control whether or not a portion of that DNA is actually turned into a protein. Right? We'll get more into the nitty-gritty details of that in, I believe it's the next unit. Another way that it can regulate whether these pathways will happen or not is just simply cells communicating with one another. So cell signaling, right? So one cell signaling that maybe it needs more of a molecule, or maybe it has too much of a molecule. The one that I'm going to elaborate on is this biochemical regulation. And this is where we have feedback inhibition. So if we have a pathway, right, maybe there's multiple steps, multiple enzymes involved, it produces a final product. When that final product accumulates to a point where we no longer need it, it's that final product that will inhibit the production in the first step of the pathway and kind of inhibit itself from being made. All right, my next slide is going to have a visual that'll help you understand this a little bit better. All right, so genes can be turned on or off. Cells can talk to one another. Or we have feedback inhibition, where the final product kind of regulates itself. So if we look at feedback inhibition here, right, we're going to look at this fictitious example. We have this pathway consisting of an initial substrate, two intermediates, a final product, and 3 enzymes that move this pathway along. Right? Enzyme 1 will take in the initial substrate, and a reaction will occur. We will now get our intermediate product. That intermediate product will be taken in by enzyme 2. A reaction will occur, and we'll get our second intermediate. Our second intermediate is taken in by enzyme 3. A reaction occurs. We get our final product. Right? Great. We want to produce this final product. But there comes to a point where we no longer need as much of that final product. We have to kind of turn this pathway off. When this final product accumulates to that level, this final product will go back to enzyme 1 and will combine with enzyme 1 in a site that we call an allosteric site. So it's not the active site, right, but rather an allosteric site. See, in this image, it's kind of opposite the active site. When it binds to the enzyme in that site, essentially that enzyme is going to change its shape. And we know shape equals function. The shape is changing. It can no longer bind with the initial substrate. It basically shuts down the pathway. Now, when we need to produce more of this final product again, this final product can be released, right, and the pathway can move along just like it did initially. There are many, many examples of this in our human body, right? Because as humans, we need to regulate so many different substances and processes. So I've designed this next slide just for you to kind of think about what you've learned so far. Ask me questions if you need to, and then answer this very easy question: what do we need to regulate in our bodies? Right? There are so many answers we could have for this. So, did any questions come up about the material that I can answer? All right, we're all pros already. I like it. So, what are some processes or substances that we need to regulate in our human body? Blood sugar, oxygen, temperature, water, salt, so many different things. I don't know about milk. Did you say milk? Oh, alcohol. I mean, your body has to regulate that too, right? Now, liver plays a huge role. So, we're regulating all of these different things, and a lot of times regulation will come in the form of feedback inhibition, and other times there's other ways the body can manage this. All right, well now let's dive into cellular respiration. Right? So, the overview of cellular respiration is it's the process by which living cells obtain energy from organic molecules. Right? It's the process by which we take in food, and that food is broken down to give us molecules of ATP and NADH. Now, part of this unit is also going to be about photosynthesis because they go hand in hand. Right? If we look at this image, we see we have our mitochondria producing our ATP, but it's also producing CO2 and water, which then chloroplasts will utilize to produce oxygen and glucose, which then the mitochondria will utilize, right? And it's this never-ending cycle. Now, a lot of times people think plants have chloroplasts, animals have mitochondria, right? But plants also have mitochondria, right? There are parts of the plants that don't photosynthesize that need to use cellular respiration as well, but it's often overshadowed by the fact that plants can photosynthesize. All right, so cellular respiration, we're obtaining energy. Okay. Our primary goal is to make ATP and NADH. Now, we're going to look at aerobic respiration, which means we are going through cellular respiration in the process. Oh, also that word is like gone from my mind. Where oxygen is also available, right? So we have oxygen. So aerobic respiration, we have oxygen, we're using oxygen. Right? We're consuming oxygen, and we'll see when we talk about the steps here, we'll eventually produce CO2 that will be released. Now this is a nice diagram of all the steps, and we're going to go through these steps. And we see that glycolysis is this first step. Now, glycolysis can also be anaerobic, which means no oxygen. Okay? So glycolysis can happen if there's oxygen and if there's not any oxygen. If there's no oxygen, it's going to produce 2 ATP, and that's the end of the process. Okay? If there is oxygen, it's going to continue in on the pathway and will eventually produce around 36 ATP. So much more ATP is produced in the presence of oxygen. As we talk about cellular respiration, we are going to talk about it in terms of glucose, right? Because it's the most common example. You see it very often, right? Other types of molecules will go through cellular respiration in slightly different variations. We're going to look at the glucose molecule. All right. So our cellular respiration, first step is glycolysis. There's actually 3 more steps afterwards that we will get to on Monday. Right? So first is glycolysis. The next step is called the breakdown of pyruvate. Then we have the citric acid cycle. Then we have oxidative phosphorylation. If we were to take these 4 pathways and generalize it into one equation, we would get glucose plus oxygen yields carbon dioxide and water. Okay, but there's many more steps in between to get to that. Now we're going to start by talking about glycolysis today. Okay? And then we'll come back to the other 3. All right, so if we look at a different way to visualize these 4 steps, we can look at where they are occurring. So it's those same 4 steps that were listed on the past slide. We have glycolysis, breakdown of pyruvate, citric acid cycle, and oxidative phosphorylation. Glycolysis is the only one of these steps that does not happen in the mitochondria. Right? It happens in the cytosol. It happens outside of the mitochondria. The rest of these steps will happen in the mitochondria, but glycolysis happens in the cytosol. And that's, I think, the big takeaway from this image at this point. All right, so glycolysis happening in the cytosol. That is stage 1 of cellular respiration. And like we mentioned, glycolysis can happen with or without oxygen. If there is no oxygen, we will end at glycolysis. If there is oxygen, we will move on to the next 3 steps. Now, glycolysis is really identical in almost all living species. It's highly conserved in its evolution, right? Meaning no matter what organism we look at, it's going to be nearly identical in every organism. Right? It's always going to contain these basic 10 steps in 3 different phases. If we look at glycolysis in humans versus the fruit fly, right? They're going to be nearly identical. Now we do have these 3 steps, right? We have energy investment, right? That should give you a clue that we're using energy, we're investing energy in this stage. We have cleavage, which is where we're breaking apart, we're cleaving apart a molecule. And then we have energy liberation, right? Where we're liberating energy, we're releasing energy from molecules. Now there's a lot going on here. We're just starting with glycolysis, and we're going to see there's a lot of steps here. There's 10 steps, right? I don't expect you to know the specifics of all 10 steps, but as we're learning cellular respiration, pay attention to any molecules that I might have bolded. These are molecules that I would expect you to know. All right, let's dive in. Stage 1, energy investment. this stage, we are using energy. We are using molecules of ATP. If we look at our diagram here, we have our glucose molecule that we're going to start to break down. Right? It goes through the first 3 steps in this phase, right? And within those 3 steps, we are using 2 molecules of ATP. Now, at the end of this first phase, we get a molecule called fructose 1,6-biphosphate. So, stage 1, energy investment stage. We are using energy, and we're turning glucose into fructose 1,6-biphosphate. Now, if we look at fructose 1,6-biphosphate, we notice it has 6 carbons. As we go into our next stage here, right, into the cleavage stage, we are cleaving this molecule apart. Right? We're going from 1 6-molecule or 6-carbon molecule into 2 3-carbon molecules. Right? So we have just created 2 molecules, right? And that molecule is called glyceraldehyde 3-phosphate. So from 1 glucose molecule, we get 1 molecule of fructose 1,6-biphosphate. Then we get 2 molecules of glyceraldehyde 3-phosphate. All right, so now we have 2 of these molecules, 2 glyceraldehyde 3-phosphate, that are going to continue on in this next phase. All right, so we have our 2 molecules. Continuing on, our last phase here is our energy liberation phase. This is the last phase of glycolysis. The last 4 steps in this 10-step series. This is where we're taking those 2 molecules of glyceraldehyde 3-phosphate. Each of those molecules are going through steps 6 through 10 on their own. Okay. In those steps, we are releasing energy. Right? This is where we get the net production of ATP and NADPH. This is where we're producing ATP. This is where we're producing our NADH. For each 1 molecule, I'm going to abbreviate this so I don't have to keep saying glyceraldehyde 3-phosphate. I'm going to abbreviate it to G3P. For each molecule of G3P, right, we produce 2 molecules of ATP and 1 molecule of NADH. Okay. Now we're thinking about this in terms of a glucose molecule. 1 glucose molecule has produced 2 G3P, right, which individually go on through these steps. So altogether, 1 molecule of glucose produces 4 ATP and 2 NADHs. Now the end product here is this molecule called pyruvate. This is the last product in glycolysis before we move pyruvate into the mitochondria and continue on with cellular respiration. All right. Glucose, using some energy to get fructose 1,6-biphosphate. 1,6-biphosphate is cleaved into 2, and we get 2 molecules of glyceraldehyde 3-phosphate, which then, through a series of reactions, turns into pyruvate and releases ATP and NADH. So in this slide here, it really just kind of combines the last 3 slides together, right? But the big takeaway here is that our net yield is only 2 ATP, right? So I said we produced 4 ATP, right? Which we do, but we use 2 of those in that first step, right? So we're only really producing 2 ATP that the cell can use. Now we're also producing producing two NADHs. Right? Not that NADH is forgotten about, and I forgot to put it on my slide. I should also say that the net yield equals 2 ATP and 2 NADHs. All right. We still have 5 minutes here. Let's answer this last question before we leave. I've given you a simplified diagram of glycolysis that's missing some of the intermediate molecules. I want you to fill in those molecules and then tell me, where does this process occur in the cell?
Right, I have listed pretty much all the homework that's due for this unit down here. You can get started on the scientific literacy assignment now. Right, you can start on the content check, but likely you won't get very far until we have class on Thursday. On Thursday, we are going to be covering a lot of detailed material on cellular respiration. So I would recommend, just to prep yourself, right, so you're in the right headspace to talk about cellular respiration, to watch this video. But it's optional.